I've noticed that there's a common theme between the three speakers today, Professor, and it's one of landscape, situation, and terrain. Professor Abou-Garbi discussed the new business models and what that means in terms of interactions with various members of the supply chain or consultants, et cetera. And Dr. Piervincenzi discussed the issues to, shall we say, the last leg, to ensure the quality and the safety of the delivered product. Mine also is very similar, but it's more focused on an area of what would be called nanomedicine or nanoparticles. I'm going to be reviewing the FDA experience, and I'm going to be talking about how categories are emerging. And once you have a category, you want to have some level of consistency across the category that leads to predictability, that leads to credibility. And this is beginning to occur, or is, shall we say, on the move in the area of nanomedicines. I'm going to be discussing the challenges that there, and I'm going to be using, I'm from the chemical industry. Welcome to the pharma industry, being like the chemical industry. I'm going to be discussing some of the issues that are in chemical registration that overlap with what's happening at the FDA in terms of new drug approvals. Before I begin, though, I just would like to point out some history. We are in Philadelphia. My artwork here was from the Barnes Foundation, the first nanoscale drug entity that was marketed in the United States, to my knowledge, was done here in Philadelphia in 1902. By Albert C. Barnes. It was a albumin stabilized colloidal silver particle. Today it would be called an albumin-bound particle. At that time, of course, the FDA was not involved in pre-market approval, so those who swallowed the drug were a part of an epidemiological study they were unaware of. And in the 1930s, they found out that the primary side effect was a permanent blue tinge to your skin called argyria. That cosmetic endpoint is still used in determining occupational exposure levels and also the amount of silver allowed in safe drinking water. This topic's going to come up a couple of times through the presentation. It shows the inventiveness. It shows how certain topics have not yet been fully resolved. It also shows the importance of improving our understanding of the technology. The FDA colleagues have published a number of articles in the last year, 18 months or so, regarding their experience with submissions in the area of complex drug products. This information is taken from an article in the Nature Nanotechnology in late April this year. And just to go through, it's a very detailed article. Those of you from the pharma industry would probably extract more information than I did in preparing this slide, but essentially they're seeing greater and greater number of submissions, INDs, new drug applications, as well as abbreviated new drug applications. They see a tendency from what they are calling reformulations for bioavailability and biodistribution. They call that the first generation. And that would be 33% of all the approvals, or excuse me, of all the submissions are liposomes, 23% are nanocrystals, and 14% are iron polymer emulsions and micelles. That represents 70% of to date, but they're now seeing a greater number of what they're calling complex microstructures and multifunctional, multi-component, meaning the drug entity itself has a geography, has a descriptive requirements, and has components. Of the 234 INDs, 44 became NDAs, 34 were approved, 15% throughput if you go by an IND starting point, which I understand is very similar to what they see in the biologic complex drugs. What's important here, I think, is that some subcategories of what are called platforms are being discussed and emerging. And I'm going to give you some of the information from another article, but relatively similar approach. This would be the nanocrystal category. Here, by the way, the time frame between the first two data points is 10 years. But there is a considerable, again, increase in number of materials. There's here more ANDAs are being approved. It's a technology that the agency is more comfortable with. Oral application is significant. Anti-infective and anti-cancer applications are there. In terms of throughput, 9 of the INDs eventually were approved. And in case of the 26 ANDAs, 8 were approved. So we're talking about the neighborhood of 20% throughput or 30%, depending on which one of those two categories you're interested in. Very often, the issue here is dissolution or solubility, and that is one of the reasons that I think that others are trying to recategorize not only nanocrystals, but the whole general field. Because, excuse me, just go back to here. The challenge, I think, is that in the nanocrystal area, they have begun seeing more of the generic translational points than they have in the other areas. They're comfortable with the general topic. They've addressed the ANDAs in terms of the generics, but they're also still finding that they're having difficulties. And I'm going to bring in the an analogous situation on the chemical registration. In toxicology and ecotoxicity, we have about a 3-year cycle time between when someone makes a statement in a journal article and when it's corrected. And I use here the example of fullerene toxicity to largemouth bass in 2004, corrected in 2007. The difficulty in chemical registration is people still cite the 2004 article by a factor of 6 or 7 to 1. In the case of the FDA regulatory perspective, though, they are able, through the guidance and through the various partnerships that occur, shall we say, to stop the loss of momentum, to go back to, shall we say, the misstatement or the misstep. But at the same time, with that experience and the ability to keep everything moving forward, they still have difficulties with proving sameness. And this has become one prompt for their evolving requirements on physical chemical characterization. So a group of colleagues, Dan Crommelin from University of Utrecht is very prominent in this, Scott McNeil here in the United States. They're trying to draw an analogy between the biologic complex drugs and which is governed by a different law, a law that also allows some, shall we say, acknowledgement of the differences in biosimilars, I mean not biosimilars, but in natural products. And they're now trying to separate, shall we say, the totality of an administrative category, meaning complex drugs, which can include things like delivery systems. They'd like to focus it more on either the carrier or the actual drug active itself. And the definition that has come up is it's not biological, it's not homomolecular, which would be the small molecule. It cannot be fully characterized, which means that the chemical or the critical quality attributes are often unknown, and they need a reliance on a very well-controlled manufacturing process. Unfortunately, everything here is what's not what it is, and there's currently a USP FDA subcommittee on really trying to provide some, call them more constructive definitions, because these are the type of materials that we're talking about in the illustration. We're talking about liposomes. We're talking about nanocrystals, meaning that there's a core material that probably has PEG on the surface for our recirculation purposes. There's an iron carbohydrate complex that there's in the bottom left. This, however, still remains an administrative categorization. Not all of these materials are nanomaterials, but there is a general tendency to say complex drug products equals nanomaterials equals nanomedicine. In some of these cases, for example, the polymer protein conjugate, it probably is a molecule. Dendrimers would be at the borderline between what those of us in the chemical registration field would call a nanomaterial. And the distinction I'm trying to draw there is the presence of an interface, an inside and an outside. More than a molecule, less than bulk. That's kind of the rule of thumb with respect to nanomaterials, and surface characteristics are very important. In trying to portray what the challenges are for this category of materials, the colleagues in this area, and by the way, there's a working group, I think it's Vifor Pharma, Teva, and also Sanofi Aventis are providing funding for it, but they're attempting to, shall we say, give qualitative direction as to what the challenges for them are. And what you have here is from the New York Academy of Sciences. There's a video capability that you can look at over the internet on this if you wish to, but they've tried to put qualitative measures of what this means in terms of moving from the innovative patented product over to the generic. What are the challenges? And also, when you do that, you have to reconsider what your original case was, what your other materials are that are in the hopper, so to speak. You're trying to drive it towards consistency. And in this case, on the bottom row, the white circles are the complex drug products that involve delivery systems. And they're not included, but the emphasis here is going to be on carriers and the drug actives. But you can see the narrow therapeutic index, which Professor Bagabria mentioned at one point. You have here the topical drugs and the oral inhaler products. and the relatively, shall we say, simpler terms of determining pharmaceutical equivalence, more difficult in terms of bioequivalence or biodistribution. The other materials are the categories that these colleagues are proposing. Two of them I'd like to bring to your attention are blue circles with respect to, and have a green, shall we say, halo. One of the issues there is that in Europe they would be considered biologics, in the United States they would be considered non-complex, non-biological. One of them, which is kind of in the middle, is albumin-bound nanoparticles. That's where the product Argerol would appear today if it were appearing as an NDA or an IND. This group has, again, an attempt to provide more definitive definitions, and I think it's Tony Hickey of RTI is leading that USP FDA effort. Now, the FDA guidance on application of nanotechnology, firstly, they don't define a nanomaterial because the Office of Science and Technology Policy directed the agencies not to define the word nanomaterial. In Europe, that took a different course, and I'll mention that momentarily. But the FDA guidance in the application of nanotechnology starts with a size range, but then shifts in their guidance to properties and phenomena attributable to size up to 1,000 nanometers. They go beyond 1,000 nanometers in their considerations, but they feel that's a well-established field. What they're looking for really here is unusual changes in properties or unexpected changes in properties below 1,000 nanometers. And I would say what we're talking about here is scrutiny, a lot of scrutiny, more scrutiny if it's below 500 nanometers, a lot more scrutiny if it's below 100 nanometers, looking for changes in properties, but properties drive functionality, so you're going to go into somewhat of a circle in terms of what this is actually meaning to you in your individual case. Not in the guidance, but in other articles that the FDA colleagues publish is the issue of surface phenomena, and they're talking about, in this case, the magnification, such as dissolution and drug release, they're expecting at the high surface to volume ratios. Again, not emphasized in the guidance, but emphasized in other publications. And what I've noticed is that the same people who are writing these summary articles of their experiences with nanomaterials are also writing articles on PK, pharmacokinetics, and physiologically based pharmacokinetic models. Now, the agency went from size to properties. There is no real connection, and one of the reasons there is that the definition of nanoscale is somewhat arbitrary. And I don't mean you make it up, but I do mean that there's a wide spectrum of sizes you can choose from and meet your mission or meet what your domain and requirements are. And what I have here is some sizes that are in various USP and other areas. 10,000 nanometers is the basis for quantifying extraneous particles. 5,000 nanometers, the upper limit for injectable drugs. Obviously, the D50, or the median size, is going to be somewhere more in the 2,000 to 3,000 size range. 1,000 is what the FDA's upper limit is to dimension-dependent properties as an application of nanotechnology. They're emphasizing properties. The House of Lords Science and Technology Committee also recommended 1,000. They were based upon biological effect. I like to always include them because we talk about competent authorities, and the House of Lords has been competent since 1254. 500 is appearing in some default modes. In Switzerland, for example, it's somewhat in around the range of the upper size of viruses, which introduces you into cell entry issues. And there's anecdotal information from Europe that this may become the size of what would be considered a bioavailable particle. 1 to 100 is the ASTM, ISO. It's also the European Commission definition. EPA and ECHA, the European Chemical Agency, are also following this. I would say that between ourselves and Europe, the European Commission proposed an interim definition to see what happens. We in the United States are seeing what happens so that we have a better definition at the very end. But it's still an area of I would say, complexity in that each group is using the definitions and size differently. Each is concerned about properties, but again, they do it from a different perspective of size. The European Commission started off with ISO definitions. I was participating in that ISO TC229, and what we did there is we defined the size range, and then we decided shape. Particle is spherical globular, fibrous is nanofiber, planar, and then we described the surface reactivity. heavily towards the chemical registration area. But if you look at ECHA or you look at the EPA, you will see them talking about nanoforms, because if you change the shape or the surface, you've created a new form. And so there's a multiplicity. Titanium dioxide can have many forms in the nanoscale range, and that's what you will see as an emphasis primarily through the European Commission's definition. This leads again to different approaches in different subfields. In the case of cosmetics, and this is a cosmetic-initiated event, colleagues in Europe were proposing nanoscale hydroxyapatite as an additive in mouthwashes and toothpaste. The Scientific Committee for Consumer Safety has responsibility in this area. I'd recommend monitoring them because of the arguments they make, translating that into your own situation with respect to drugs. But these colleagues decided that these materials had needles in them, they had spheres, they were in the nanoscale range, meaning 1 to 100. There was insufficient information for them to make a decision regarding safety. It happens at about the same time, colleagues at Arizona State University looked at infant formula and a number of food materials. They also found the same nanoscale materials and needles as presumably soon to be grass, yet we have the agency's admonition in 2014 not to assume that grass carries over to small materials, and therefore you should have some nanoscale testing. Again, this is a situation where having a definition in Europe is creating a situation of people scrutinizing what they have, asking questions. It shows progress in understanding, but it also leads to more immediate questions right now. Similarly, EFSA is responsible over in Europe for food additives. We had an academic article in 2012, and I'm picking up, by the way, here a 3-year cycle time in terms of action and then a reaction in the general, shall we say, journal article area. But a 2012 academic article on E171, which is food-grade titanium dioxide, they observed 36% particles below 100 nanometers. The EC definition is 50% as a threshold. So these authors looked at what that meant in terms of dietary exposure and decided that children may be disproportionately exposed. In 2015, there were 4 articles from industry. The EFSA group was going through a scheduled review of E171. The industry commented that it was only 21% by number, and then had a number of, shall we say, I would say, undiplomatic commentary on the naivete of the academics who had talked earlier. They didn't use a OECD technical guide test. They weren't certain of this and that and so forth. What's intriguing here is that the industry people who wrote this article did not sell E171, though they had done the TG from OECD and they had actually done excellent GLP work. You see some of the differences that occur as different actors make their arguments. The EFSA review renewed the E171 approval, but they also added that there was missing data because now that Nano has come along, their understanding and their questions have increased. There's actually a data call out right now, will be finished in June, asking industry to come up with more results of various tests, et cetera. Now they're concerned with the alumina coating that's on the titanium dioxide surface. Evidently, you can exceed your daily allowance of alumina just through the titanium dioxide that is put in various colorants such as skim milk. My point here is that definitions lead to scrutiny. Scrutiny leads to questions and better understanding, and I'm certain that the colleagues who are recommending the non-biological complex drug products be better defined or more actively defined are also anticipating that there'll therefore be greater scrutiny and there'll also be more questions, but the finding also in the long run greater consistency, greater predictability, and therefore greater credibility for the products. Returning to FDA, this comes from another article. Catherine Tyner is very prolific in this area. These are what they call platforms. I've been calling them categories. This is not the first of this category, but it is, shall we say, a prototypical one. Provided here the date of the approval of the NDA and also the approval of the ANDA. That's going to be to the generic sort of version. 10, 12 I'm saying that the cycle time in terms of this effort is more in the 10 to 15 time range, meaning when the FDA reconsiders its original approvals and has to address some of the issues of how do we define sameness. There's an inorganic nanoparticle separated from nanocrystal. The arginol example, as used from Dr. Barnes, would actually fit protein-based as well. And I point out that the Abraxane was first introduced to avoid the nausea that was in the solvent system. That was the reasoning that Dr. Barnes had in 1902 for his product. So we're revisiting some of these themes or some of these topics on a regular basis. I participated about a month and a half ago at a USP-sponsored workshop in nanomedicine, and I'm a physical chemist observer somewhat, watching my colleagues wrestle with their problems. I'm very calm and collected about the FDA, but if you ask me questions about the EPA, I'm more involved with what they should be doing. So mine was more a disinterested observation of my colleagues, and what I noticed was somewhat of a split discussion going on. One is that normally critical quality attributes are based upon a full phys-chem characterization of the material so that you connect your manufacturing capabilities to what your product's going to be. And then separately, physical chem characterization is useful for the PK/ADME determination so that you guarantee or ensure that there's therapy and toxicity that you've monitored in the laboratory in your various clinical trials. If you have difficulties with the phys-chem properties and them being incomplete, now you do not have a connection really between your critical quality attributes and your PK/ADME expectations. This also means that you may be moving from a kinetic limitation of dissolution rate to a solubility limit, but how do you analyze that for deciding what that situation should be? That's also used with your sink and non-sink type of conditions. What I've noticed in the chemical registration arena is we have a similar problem, but it's called, if you have a metal or a metal oxide, was the toxicity due to the particle or was the toxicity due to the solubilized metal? So zinc oxide is the example. Toxicity to the particle of zinc oxide, or was it toxicity to the zinc ions that were released in shall we say, during the test measurement or in the body? I'm back somewhat to arginol because this is recent results using silver and fetal bovine serum albumin. But what the colleagues have noticed is that the more FBS present in the system, the more the solubility that they're measuring of the silver. Now, in normal discussions with regulators present, everyone starts arguing about what the right test condition should be, what the standard should be, trying to find one test or one set of conditions that should be descriptive of all possible particles. And these colleagues were, I think, very unique in that they treated this as in vitro release. And they actually were able to measure the difference between if you put the particle in the RPMI buffer first and then added the FBS, you got a different answer. They also looked at 3 different manufacturing techniques for making these silver particles, and they saw differences, again, in what I would call an in vitro release. So what I'm perceiving from the chemical registration side and what I also saw in the nanomedicine workshop was an attempt to, shall we go, from the critical quality attribute, measuring production, over to a PK direct, and forget about or not worry about the incompleteness of physical-chemical characterization in between. These colleagues are doing the same, but in the nanoparticle ecotoxicity regime. I'm mentioning this or highlighting it because what is happening in Europe right now is that there was a large FP7 project called NanoReg. It had connections directly with the regulators. It was supposed to answer their questions using about 18 commercial materials. And they are now have their final report, and what they're recommending is 17 tests being changed. They actually are talking about 13, 14 tests being changed and 4 new ones being added to the OECD technical guidance. I think that they're fighting the situation of what they were looking at in these individual tests, and that would mean they'll move over more to what I call the in vitro release approach, because that would be more specific to manufacture and production and less on the screening side. But this is an area that is being recommended, and each of these groups will require different levels of interlaboratory type of guidance or studies to be certain that these are appropriate, these various modifications. This is being proposed. The OECD meeting is actually tomorrow in Paris, where they'll be pursued and presented. If you propose a modification, that means you have to communicate the modification, you have to test the modification, you have to be certain it's valid. And what I'm perceiving in a number of situations informs that colleagues at FDA are recommending that people participate in various levels of standard developing organizations. Standardization activities in general, I like the definition that Dr. Persigenzi offered, which was it's not necessarily the ASTM standard, but some level of approval or collective wisdom on this. I mentioned earlier the FDA USP subcommittee, ISO TC 229 mentioned their definitions. Their European colleagues are very active in that area and often use that as a forum through which to propose various test modifications. Kind of a bubbled up from the roots, shall we say, bottoms up was nanoinformatics, data and modeling. I noticed again that that was mentioned in terms of data systems. It's called nanoinformatics, but to bring into your attention that there is a work going on for the Nanoinformatics 2030 roadmap. That is an attempt to find how to set up a database, how to curate the information, what the ontology should be, so that you can use various modeling approaches to actually make predictions. It's a reliability, quality of the data, so that you can see the differences in terms of PK modeling. That is Dr. Andrea Haase from Germany is leading that. The US input is going through the US-EU Nano-EHS group. NanoReg proposed database issues. The CA Nanolab, which is associated with the Nanotechnology Characterization Laboratory, they have a similar effort going on in terms of how to structure a database. They actually have a public database with 1,200 entries, various products, various materials for the purposes of therapeutics, and especially in the cancer area. And there's also a weekly effort called the Nano Working Group that is NIH, NCI oriented. Besides these, ASTM E56, the more traditional standards developing organization based here in Philadelphia, or actually up in West Conshohocken, they have an informatics standard, which is called ISOTAB Nano. Colleagues in Europe have one that's called ISOTAB JSON. The attempt here is to connect these sorts of materials to the wider world of bioinformatics. We're having ongoing balloting on a uniform description system, which came from CODATA, which is one of the basic units back in Paris. The International Union of Pure and Applied Chemistry would be a CODATA type organization, but they are working on this and how would you describe a material effectively. And also more recently, and through some prompting from FDA colleagues, they are forming a nano-enabled medical projects committee, where topics will include characterization, but also going back to this in vitro release concept, I would call them more the functional assays for stability of these materials, and finally performance of medical devices that use nanomaterials. And I believe this is more the biosensor area than a material of construction. In summary, the FDA's case-by-case experience starting in the 1970s has built up a body of work, body of knowledge that's allowing them to identify platforms, subcategories as their phrase, useful for a consistent approach so they can communicate that consistency to the general public and to the pharma industries in particular. Demonstrating sameness remains the challenge, and implies that there should be early and sustained involvement of manufacturing throughout development. And I don't mean just that I can make what you characterized. I mean, what is my in-process control? What is my ability to control the envelope, shall we say, of the manufacturing process? What does that mean in terms of PK and ADME? Characteristics would be an example. And also that we can anticipate modifications to established test methods where the SDOs and other organizations act as, shall we say, resources for communicating the need for the change and validating the change to some level that it can be more prominent and be more useful to the industry as a whole. And with that, thank you very much, and would of course take any questions.